Presented by Caltech.
What I want to tell you about, I think many of you know that neuroscience is big at Caltech. There are faculty all across the campus who are addressing the amazing problem of the nature of the human brain. And we're a small part of that. I'm a chemist, so I'm a molecule person. So when we talk about chemistry on the brain, we talk about thinking about the molecules of the brain. And what I'd like to tell you about quickly is how we figured out a way to do chemistry on the brain and what we've been able to learn from that, including unraveling a fundamental mystery about the way nicotine affects the human body. So to get to the chemistry on the brain is not that hard because it turns out the fundamental signaling system of the nervous system is chemical in nature. A neuron on excitation releases a small molecule, the neurotransmitter, and it drifts across the gap between two neurons called the synapse, where it is then picked up by receptors and the signal is reprocessed. So the signaling of the nervous system is fundamentally chemical. And the synapse is complicated, and the brain is really complicated. But we're chemists, we're molecular, we have to get right to the molecular level. So let's look again at a simplified synapse. And proteins on the surface of the neuron process the information. Again, the neurotransmitter drifts across the synapse, where it binds to a protein shown in the upper right, embedded in the membrane because it's on the surface of the cell, and it's a protein with a hole down the middle, but normally the hole is blocked. But when the neurotransmitter binds to the protein, it induces a large structural change in the protein that opens the channel. At that point, charged particles, ions, flow across the cell membrane. And the flow of ions across a barrier is identically equivalent to an electrical signal, and that's the signaling mechanism of the nervous system. This is the process we'd like to understand in detail because these are fascinating molecules. Let's look a little closer at the players. Here's one of the receptors. It's called the acetylcholine receptor because acetylcholine, that small molecule on the right, is the neurotransmitter. Just 7 carbons and some oxygen and some nitrogens. It's a little molecule, and it's the signaling molecule. On the left is the receptor, the protein that's, again, embedded in the cell membrane. It's a much bigger molecule. You can see it's a nanostructure, 12 nanometers by 7 nanometers. To give you a sense of the scale, if you're not completely calibrated, on this receptor, I've drawn acetylcholine to scale on that slide. It's right there. So that little molecule binds to that molecular behemoth and launches a signaling cascade. That's what we want to understand. Now, it turns out the acetylcholine receptor is actually a family of receptors, 20 or 30 very, very closely related but subtly different receptors found in different regions of the body that all do basically the same thing but in subtly different ways. It plays a prominent role in the brain where it's involved in memory and learning and cognition. It also controls the neuromuscular junction. Every time you move a muscle voluntarily, it's because acetylcholine was released from a nerve, it drifted across the synapse and bound to the receptor in your muscle. Ions flow, muscle contract. Okay? So it's important in the central and peripheral nervous system. This is the longest known, best studied receptor of any kind, and it's quite famous because nicotine activates this receptor. In fact, there are regions of your brain that are more sensitive to nicotine than they are to acetylcholine, but it's an acetylcholine receptor. Nevertheless, this feature is so prominent that most people refer to this as the nicotinic receptor. So we actually work on the nicotinic receptor, but it's an acetylcholine receptor. Remember, it evolved to bind acetylcholine. So it is a certainty that the initial chemical event of nicotine addiction is nicotine binding to and activating this receptor in the brain. And we know the consequences of that are not positive due to deaths caused by smoking. On the other hand, nicotinic receptors are established therapeutic for a host of neurological disorders. For example, there's many twin studies that show that smoking is protective for Parkinson's disease. So the Michael J. Fox Foundation has a clinical trial right now for the nicotine patch to see if it slows the progression of disease. So these are important therapeutic and pharmaceutical targets, and again, we want to understand how the chemistry happens. And also, if you think a minute about everything we've said so far, it brings to light a chemical mystery about the action of nicotine. Remember, it's active in the brain, addiction, but it's also active at the neuromuscular junction. So the question is, how can smokers tolerate nicotine? Why aren't they twitching all the time, or paralyzed, or worse? And so it's very much a chemical question. There are very, very subtle differences between the brain and the muscle receptor, and we want to understand how it is that nicotine can cause you to become addicted but not paralyzed. It's a chemistry question. It's a molecular scale question. We need chemical tools to address it. And the molecules of neuroscience are complicated, so how do we develop those chemical tools? Well, what we've done here at Caltech is hijack the genetic code for our purposes. Remember, proteins are made up of a string of 20 amino acids. Amino acids are the building blocks of proteins, and the receptor's a protein, and there's 20 amino acids that string them together. Now, it turns out, to a chemist, 20 is not a very big number. We can make amino acids in the lab, and we can make thousands of them, tens of thousands of them. That's what we're used to. Now, I guess 20 is okay if all you want to do is create all known life on the planet. But to a chemist, it's very limiting. Like I said, we can make thousands of amino acids, and they would give us the power to probe these things as if they were real chemical structures. The problem is the molecular synthesis machinery of the cell won't incorporate these amino acids into proteins the way you want to. So that's what we had to develop at Caltech, was a way to hijack the genetic code so that would happen. And so here at Caltech, as part of a long and fruitful collaboration between my lab and Henry Lester's lab in biology, with early help from John Abelson and the great Norman Davidson, right? In chemistry, we're celebrating the leadership chair, the Norman Davidson Leadership Chair, and this is the 100th anniversary of Norman's birth. That group got together and created a new way to incorporate unnatural amino acids into proteins anywhere we want, anyhow we want. So we don't just have 20 amino acids, we have over 120 amino acids. What this lets us do is think and act like a chemist. It gives us the ability to ask really high precision questions about very complicated molecules, but really think in detail about what's going on. So, what have we learned? Let's look at 3 molecules. Again, here's acetylcholine, we've seen before. There's nicotine. One more molecule. This is varenicline, which is marketed by Pfizer as Chantix. You've all seen the TV commercials. This is Pfizer's orally available smoking cessation compound. And of course, was developed to target nicotinic receptors. The first commercial oral smoking cessation compound. So, a lot of studies by many groups, including our own, have come up with a model for how we think these drugs interact with the protein. There's 2 key parts. One is over on the left there, the nitrogen of nicotine, the nitrogen of varenicline, and the oxygen of acetylcholine. They are thought to make a hydrogen bond with the protein. So the NH is part of the protein. The nitrogen and nicotine makes a special but very common interaction called a hydrogen bond that's proposed to be important. The other important part is over on the right there, the positively charged nitrogen, right? It's a cation. Positive charges are cations. And everybody knows how you bind cations. Opposites attract. You bind positive charges with negative charges. So you have a cation, and then the negative charge is called an anion. And 2 of the 20 natural amino acids are anions, so the protein could put an anion next to the positive charge and obviously bind it. So again, only the unnatural amino acid methodology lets us think about interactions at this level. So we went after them. First, the hydrogen bond. What did we find? Well, it turns out acetylcholine and nicotine make a really good hydrogen bond. It's just like we thought. But then we got a surprise. Chantix doesn't make that hydrogen bond. Now, we know for a fact that the scientists at Pfizer, when they were developing this compound, they put that nitrogen there so that it would make the hydrogen bond that nicotine makes. But it actually doesn't. It's no effect at all. So it makes you have to rethink, if you're trying to develop new drugs that target this receptor for Parkinson's or Alzheimer's or schizophrenia, it makes you rethink how things have to work. What about the other part, the positively charged nitrogen? Again, it's going to be a negative charge, right? Except, out of the roughly 2,500 amino acids that occur in the protein, in the receptor, we know exactly which one binds the positive charge, and it's not an anion. It's not a negative charge. Instead, the binding of the positive charge occurs by a different interaction that was discovered in our labs at Caltech a while back called the cation-pi interaction. So instead of cation-anion, the cation binds to a system that is a special kind of chemical bonding called pi bonding. 3 of the natural amino acids have that pi bonding, and I think much to the surprise of biologists, but the chemists figured this out, this novel binding interaction can take place, and that's how the binding occurs. We've studied dozens of molecules in dozens of receptors, and if there's a positive charge, there's a cation-pi interaction, and it's a universal binding interaction, except there's one exception. There's one case where there isn't a cation-pi interaction, and it's nicotine at the neuromuscular junction. In the brain, nicotine makes a really strong cation-pi interaction, but at the muscle, it's gone because the receptor has a very subtle change to it that you never would have seen this unless we did the experiment. So in effect, using chemistry, we've solved the mystery. We now know why you can become addicted to nicotine, it's active in the brain, but not paralyzed by nicotine, the neuromuscular junction, and therefore you can smoke and have all those adverse effects. And it's the cation-pi interaction that causes that to happen. So this work has been done over an extended period by an amazing group of young people, all Caltech grad students. They're the most amazing young people in the world. This is pictures of them over the years. They do all the work, they do all the thinking, very much in their debt. Thank you very much.